Okay, another little session for us on the microscope. Now, hopefully we're going to understand this as it goes through and see that it is important to be able to use this effectively as we go through. All right, today I'm concentrating on magnification and also resolution. Resolution means clarity. So far we've been looking at a microscope that has the arm. We've learnt the safety reasons of handling this. We should always grab here and have our hand under here as we move it. Now we do have some microscopes. We still have the mirror and we have to take a light with us when we use those, but most of ours have the light built in now. Alright, so as we're going through, let's look at it. So here's the part that's the important part. So let's have a look at this. There are lots and lots of labels here. I'll let you read those at your leisure, and you should have pretty much this picture as it's going on. Right, keep remembering that we are using light. So a light must be able to go through the material you are looking at. And that is super important. If it can't go through when you are making these slides, as you'll be asked to do, then we have a problem. If the light can't come through them, we can't see what's going on inside. Alright, so let's start off with the idea of making it larger. Now, there are two parts to a microscope which are involved in magnifying. The first part is this part here, and that is the eyepiece. Now the eyepiece is able to magnify quite a lot. So we're talking about this part here. I'm going to go through and use the pen. Alright, let's make it blue. It's this part here. Now it can be removed, but please do not. We can damage these. Lenses are really, really carefully made, and we've got to keep them clean. So this part up the top where we have the eyepiece has to be super, super clean. And so do these lenses down here. They're the secondary parts. Alright, so the eyepiece though makes everything 10 times larger. And that's really, really good. It's sort of like a magnifying glass. Now to help us out with that, we then go down to these down the bottom. And they vary in length. You can see there are two here that are shown. They've got different length. Normally the shorter the lens, right? The shorter the lens, the lower the magnification. Now this one will probably be the red. This is how we're supposed to pack it away. So the red lens was there, and it is 4 times. So combined we have 10 times here and 4 times here. So if I use the red lens only, I am making what I'm seeing below 40 times bigger. Alright. Now the other ones have got little colours on them. This helps us, as well as the length, it's got colours on them. So as we're doing this, we notice that the yellow one, and it says here is 10 times larger, it sure is. So that means I have 10 times here and 10 times here, and I have to move that around. You have to move it around and click it into place. It must be clicked into place, and you'll understand that when you use it. Otherwise you won't be seeing too much at all. Alright, now when we're doing the focusing, we always start off with the lowest number, the red one, and we focus it in. Now when I'm normally focusing mine, I don't hold my head to here and keep looking at it as I'm going through. I don't look at the eyepiece. I basically use my adjustment, the big one, the coarse adjustment, bit more about that in a minute, to wind it down so it is about 1 centimetre above the slide. And then what I do is I simply put my eye then over it, and if I then move my lens away a little bit from the stage where the slide is, I tend to find I focus it. That's just the way I do it on the way through. Alright, what I wanted to get out of this, you've got 2 parts and double magnification. This thing is about, and unfortunately these little arrows are locked, when we do this we are looking at two parts. If I want the stage to move quickly and over a long distance, I use this one, the coarse focusing knob. Now with ours on the newer microscopes you're using, the coarse and the fine are joined together. The coarse is the bigger wheel. You'll notice you have to move. The fine is the smaller wheel on the edge of it, on the inside of it. Now when you are doing your focusing, we always use a coarse knob first to bring it in, and I talked to you just a minute ago about how I would bring it down to about 1 centimetre above on the red. Right? Then, once I've got it focused, I'll use the fine to help me focus as well. Once I've got it focused and it's ready for my eyes, I then can shift this around, the yellow one around, until I hear it click, and then use the fine one only. No need to use the coarse one anymore. So we use this one down the bottom, this one, or the inside one on our other ones. And we just focus it in. Now be very, very careful when you're doing that. Now we have got a third lens on the bottom, which makes it 40 times larger, so total of 400 times larger. If you use the coarse knob with that one, you will quickly find you can't find the cell anymore. So only ever use the fine adjustment for that one. Right, more later.